Hello, everybody. My name is Marie, and my presentation or lesson will be on the pathway for sound transduction. And hearing is the neural perception of sound energy. And what we're really doing is just transducing or changing sound in air, traveling vibrations of air, high and low pressure systems. And we're going to change that into the electricity in the brain that we are going to perceive as being sound. But in between, we are going to have some other changes. So it goes from air to being changed into the energy in the liquid in the air, which will move little hairs, and then action potentials, graded potentials that become action potentials. And the auditory cortex, we have one on the left and right sides, lobes of the brain, are made up of the primary auditory cortex and this association area underneath it. Here is a diagram of the ear, and the best way to really learn this is to start with the outer ear up here on the left side. We have an outer ear, middle ear area, and inner ear. And the outer ear, this is an auditory meatus, the pinna, which serves to gather sound waves. And there are two aspects to sound that are really important for us for our perception, and that is what is the sound and where is it coming from? And it actually takes two ears for us to figure out the exact location as to where sound is coming from. And that's an important evolutionary adaptation for us. Sound waves are going to come in through the pinna and go through the auditory canal, and then these sound waves hit the tympanic membrane. And we have to remember that because sound waves are being converted from air to liquid, that pressure changes and it's going to have to be amplified. And our bodies do that in two ways. We amplify the sound waves by the tympanic membrane. It's actually quite a bit larger than the oval window. So here's the tympanic membrane, here's the oval window, quite a big size difference. So that surface area being much larger serves to amplify the sound. And the lever action of three little bones in here called the malleus, incus, and stapes, which are three of the smallest bones in the body. The eustachian tube runs down here. That goes into the throat area, the pharynx, and then that energy starts to move fluid that's in this snail-shaped organ called the cochlea. And it's the movement of these three little bones here that are pushing against this oval window. And we're going to see that the sound and the fluid gets changed in the cochlea so that those hairs move and then graded potentials are created. The semicircular canals up here serve to help us keep equilibrium. People have problems with vertigo if you have problems with your semicircular canals. So again, that tympanic membrane, this is the area if you've ever flown in an airplane and you feel pressure changes. We usually feel that pop a little bit if you yawn or if you're chewing, and we need to do that when there are external pressure changes. This is a little cross section of where the cochlea runs through, and really the most important part of the cochlea is this organ of Corti, which are where the little hairs are. And then the organ of Corti, which, by the way, was discovered by Alfonso Corti, an Italian anatomist, who figured out that it's these little hairs here that are hitting upon this tectorial membrane, and that's what's going to cause the graded potentials. Here is a closer look of the cochlea, and it's best learned if you unravel it. So that snail-shaped organ is unraveled here. And what's happening is we have sound waves coming through, and the sound is going to take one of two routes. Either the sound is going to get dissipated, which it needs to do, or it's going to stimulate those little hairs and it's going to initiate some receptor graded potential. Here on the right, we have just a little reminder in graph form that these are patterns of vibration along the basilar membrane for these different frequencies. And we have 6,000, 1,000, and 100 hertz. So you should be able to conclude that the closer it is to the oval window, the higher the frequency. Higher frequency, closer the vibration to the oval window. Along this little basilar membrane, we're going to take a look in a little bit more detail as to what the little hairs are doing. There are 4 rows of hairs. There's an inner layer that allows us to hear, whereas the other 3 layers are just to enhance the response of the inner cells. Different sound waves strike different regions of these hairs. And this is what one would be like, kind of magnified in cartoon form. And it's sort of like striking a piano. So we have 15,000 hairs in each of our cochlea, and this is what's going to be moving. There's going to be a mechanical movement here of the hairs from the fluid because it's attached to the tectorial membrane, which is not shown here, but it would be laying right up top. So it's kind of like the little hairs have hairs on them. They're not technically cilia. We'll just call them hairs. So when there's no bending of hairs, we can see that you can have signals being transferred in action potentials, but when they move either toward the longer hairs or the shorter hairs, that is when you get either hyperpolarization or depolarization. And these little inner layers of hairs act like really kind of like a piano, and when the hammer hits certain strings, you're going to get different notes. The functional organization of the auditory cortex is complex and not really well understood. And we do have the ability to have many, many inputs and outputs, and our brains are very good at determining what those sounds are going to be. And when we talk about sound, we can be talking about harmony, we could be talking about pitch, rhythm, any of those things. On the tops of these hairs are what are called little tip links, which are little mechanical connections. And what's going to happen is when they start to bend a certain way, if they bend toward the longer of the hairs, more potassium will actually enter into the cell and cause a depolarization, which is the opposite of what we're usually thinking when we think of action potentials. But you can see that there's a depolarization where it's getting a little bit more positive, and depending on how many stimuli you're getting here, how loud the sound is, it's going to cause depolarizations to occur. Whereas if the little hairs are bending toward the shorter area, there's no potassium, no calcium released, and it's actually going to hyperpolarize and go into the more negative region, and then that is not going to be contributing to an action potential. Here is one hearing study that I found called Neural Tracking of the Musical Beat is Enhanced by Low Frequency Sounds. And this was a really interesting study where they were trying to figure out whether measured cortical activity in the brain can be represented internally when rhythm, and specifically low tone rhythms, are applied. And what they did was they took 14 people and they used EEG, electroencephalogram, wires on their heads, and they did cochlear models to try to figure out whether bass sounds really do have an effect. And it is known that humans have a particular entrainment to rhythm, and it's unclear as to really why, but all cultures, all people do it. We tap our feet, we tap our fingers, we bob our heads. There are certain sounds that we like. And again, we could be looking at different aspects of sound, but in this particular study, they were looking at bass sounds. So 14 subjects had rhythms that were played to them continuously in a repeated form. And here we can see that there was no real difference in the low tone and high tones in the cochlear model. However, with the low tones, there was a higher frequency than the high tones, and that showed up on the EEG. The unsyncopated and syncopated rhythms are just to show how the beats were actually running. And syncopation is when we have beats that we do not expect. So when we hear jazz music, a lot of times that's not like a very regular, evenly spaced beat. You get beats where they are unexpected. So this is a spectra of the acoustic stimuli, and these shaded regions do show standard error. Here are two other graphs, some results. They finally concluded that the rhythmic stimulation by bass sounds leads to enhanced neural representation of the beat and meter. And here we can see the meter-related versus meter-unrelated frequencies, and it appears that there is more activity in the low tones as opposed to the high tones, and more so in the unsyncopated than the syncopated rhythms. Here are some graphs, again, showing the unsyncopated and syncopated, and the starred areas here show that these are connections between each particular individual, what their responses were. And they noticed that these are more significant with the starred areas, with the low tones as opposed to the high tones. You might be asking, are there any practical uses for this research or why anybody would really want to do this? And there is. There is an increasing interest in using music for assisted practices for clinical rehab of cognitive and motor disorders, people who have brain damage, people who have strokes, cerebral palsy, schizophrenia, and it is used for rehabilitation of some of those conditions. They also think that there could be a connection between auditory temporal encoding for sounds like this and our evolution. I'd like to leave you with some music that really shows some bass tones and lower tones. This is an African percussion, and as we play this, I will show you my references. Thank you.